as Tom said, I'm a member of the Hybrid Ag team, and we're excited to be part of today's webinar. And our little bit will be nutritional influences on crop load. So if we go to our next slide, I'll just do a quick little intro for Joe, who's also going to be part of the show. So Joe works with us, and he works basically in 3 crops with me as well, based around pine fruit, stone fruit, potatoes, berries, and nuts and citrus. So he's pretty broad knowledge base there. So he'll certainly help us along the way. All right, so we'll move just a little bit about hybrid ag. So we're on a bit of a journey to understand more about how we can transform food and have a positive impact on human health. That's our purpose, and we're trying to transform food for health. We believe that's the starting point, and we're on this journey to discover more about what makes food truly nutrient dense. And we've begun collaborating with researchers all over the globe to try and pull that research together, which has been around for the last 100 years, to try and enhance the industry's understanding of soil and plant nutrition. So that's what we're really trying to do, is pull that together in a meaningful way that growers can truly actually have that extension out in field and make some really good use of it. And that's probably where we're trying to start today. So we'll move on to our first slide, which shows the stages of plant nutrient importance. So today we're trying to focus on some of these key stages of how nutrients can play an important role in the crop load. So what we'll try and look at is how to maximise cell density in each piece of fruit which we produce through that cell division stage, how to ensure controlled cell expansion through the fruit fill and sizing stage, and trying to achieve our target size and the requirements without compromising our fruit quality. We'll look at what impacts nutritional inputs have close to ripening as well, because there's some other things at play there. And the other thing which needs to be mentioned is we'll also look at the bud initiation phase, which is going on around that same time as ripening. So once again, you know, our brief is an overview of activities through this stage, where we can influence the outcomes through the tree and subsequently the fruit. Also, there'll be a hybrid ag series of webinars which we're planning. We've already just done one. So if you want some more information in a little bit more depth, just keep your eyes open for those as they come up. So I'll now hand over to Joe. He's going to go through and explain the cell division phase.
Good afternoon, everyone. So once we've had obviously we have flowering at the beginning of the season, and we have pollination. So as Russell actually mentioned, we have a stage known as cell division very, very closely after pollination. It actually starts almost immediately after that successful pollination, and it only lasts for a very short amount of time. So we've tried to just summarize a couple of the more important sections of where nutrition can play a fair influence. And so this is probably one of the more critical areas in the season as to fruit quality, particularly. So and also size and density. So during this stage here, we have every cell that will be in the final piece of fruit is actually created during this stage. So the important outcome here is to have the maximum number of cells created within this window as we possibly can, because it's almost the decider. It is the decider of the number of cells that you will have in that piece of fruit, which is directly related to, you know, your fruit size, firmness, which comes from obviously the density of that piece of fruit, and it all starts right at this stage here. So having the most amount of cells possible means that we can grow larger pieces of fruit without compromising the firmness and shelf life, basically. So probably one of the first nutrients, and probably one of the most discussed nutrients around fruit crops, is calcium. And so most of us will understand how critical calcium is to the cell wall and the function of the cell wall in the fruit. It's a fairly commonly discussed nutrient. But one thing to realise probably is, although healthy trees with good soil available calcium will continue to absorb calcium throughout the whole season, it's during this particular stage here that when all of these cells are created for the fruit, that calcium is at one of its most critical points. So we really need to emphasize how early we have calcium availability in the tree. And interestingly, as we heard a bit earlier, there's actually some studies that estimate that 50% of the crop's cell division is already completed by the stage that we commonly recognise as full bloom. So this is a picture we actually took yesterday, just of some cherries, just a young block, so there's not a whole lot of, not a huge amount of bud on there, but there's flowers already. And you can see we've got some of these flowers will have already pollinated. So in this particular, just to highlight how early this cell division stage starts, is within the next few days, these cherries here will have begun the cell division process. And then by the time we actually recognise full bloom in this block here, a large number of these flowers will be will have already completed, or up to 50% of them could have already completed their cell division process. So it's very, very early, and a lot of calcium is used, or a large amount of the draw of calcium for the crop is used during this stage for creating all those cell walls. So it's very, very important to have it there, because if we don't have it during flowering and during cell division, we rarely ever can we make up that calcium deficiency later in the season. It just doesn't, you can't supplement enough really, because it wasn't there in that crucial stage. So there's a number of other factors also that can impact or shorten the cell division window, which is probably most typically environment, sudden heat or cold. Applications of your plant growth regulators can influence the length and the effectiveness of that period. Pest and disease also can sometimes impact it. Water availability is a really big one, and then there's a number of other nutrients as well, which play a key role in making sure this process actually happens. So we've just got some of the key nutrients here for during cell division. So the way we've probably just structured this presentation is just we'll have some of the stages, and then we'll just have the key nutrients that need to be probably front of mind for ensuring that these are available after each stage, or during each stage. So with cell division, one of the first ones here is actually manganese, trace element. So its key role during this particular stage is actually in the regulation of potassium, or any of the cations actually, but potassium particularly in the sap flow. So we understand that calcium and potassium have a particular antagonistic relationship with each other, and in a lot of cases we can have, if we have too much potassium available in the sap in the plant during this period, we end up with a deficiency of calcium in the fruit. And the research recently indicates that the plant needs manganese to be able to control the various concentrations of cations in the sap. So we need to have manganese there for that purpose. It's also pretty important later on, and we'll touch on it again later, for water hydrolysis. Zinc is pretty critical in the cell wall, plays a critical role in the cell wall maintenance. And so the interesting side effect of that is that you actually have better resilience to environmental and pest and disease impacts on the fruit at a cellular level. So making sure we have effective or the required levels of zinc, and that goes with calcium, and then which we've touched on, and then boron as well, which is well understood for its relationship with calcium. So another one that's probably not so talked about so much is cobalt, and when we were talking about the plant growth regulators, such as mimicking hormones in the plant, such as cytokinin, and most of the cytokinin that is naturally produced in the plant is actually produced in a growing root tip. So cobalt is actually required to stimulate the growth of root tips and continual growth of root tip, and so it's actually critical to have cobalt in the plant. Although it's only required a very small amount, it's actually a very, very critical part to ensure the plant can produce cytokinins on its own as well. And then copper, very, very close related to, or because of the role that it plays in lignin production, it has an influence on the elasticity of each cell, and so allowing the fruit to actually expand and contract. And a practical example of this is in rain. Later in the season, we often see where we have better copper levels in the sap, we actually have better resilience to cracking and splitting later on. So that's an interesting one. There's a lot more research still being done into the other factors nutritionally that go into that, but that is a key nutrient to make sure it's available. One of the important things to understand here is in stone fruit and cherries alike, we often assume that we put enough copper on as fungicides. And from our research, using some newer technology, sap testing methods, we've actually found in a lot of cases that we have copper deficiencies in the sap, which is showing to us more and more that, and it makes sense, that the coppers that we're using for fungicides are purely contact, and they're not designed to get into the tree at all, generally. So in a lot of cases, we actually have deficiencies of copper and should be using a little bit nutritionally. So I've got a nice slide here where Andrew's just going to give us a practical example of cell division and its effect on cherry size.
Okay, thanks. So as you can see here, we've got a picture of a cherry cell which is on top of the screen there. We can see that it's going through its cell division phase, so it's trying to multiply. So effectively, we're in that reproduction phase or cell expansion, before cell expansion. So we're trying to multiply as many cells as we possibly can. So this is the particular part that we're trying to influence, particularly with the nutrients based around the availability of the calcium, as what Joe just mentioned, to try and maximise that cell division stage. So then the various differences between the larger and the smaller cherries, which you see left and right, is due to more or less cells which has been produced through that cell division stage. So the other really good outcome of having more cells in the actual piece of fruit is obviously the first one is you can maximise your size, but you also can maximise the actual fruit firmness when it comes to harvest time, which will also give you the benefits of shelf life and fruit storage ability, particularly if you're looking to move into export markets or in, say, an apple type world, if you're trying to do longer term storage. So you can see here, as a grower, you can really influence the outcome of your fruit quality, which is really going on so very early in the process, from that full bloom to that 14-day period post bloom where that cell division is really taking place. So yeah, we really need to think about how we can maximise that cell division stage, because it really does have a benefit on the outcome of the fruit. Because as we've mentioned, we can't add more cells to the fruit once we get past that stage. So immediately after cell division, the tree shifts into what we call a growth stage or cell expansion stage, fruit fill. So this is obviously, it's a different process in the tree. It's got slightly different hormone shift to more of an auxin domination type part of the tree. So once again, we're trying to maximise the sugar transportation into the fruit. And studies have shown that sugar within the fruit has a direct correlation to the dry matter content. So this means when we can actually achieve abundant sugar levels in the fruit, we'll certainly have better size and once again, better fruit weights at harvest. So the sugars or proteins are produced in the leaves, and this is obviously the process which is called photosynthesis. So I'll probably just hand back to Joe and he can just go through this photosynthesis in a little bit more in depth. So this is what's taking place, and we really need to try and maximise this photosynthesis process to get the best fruit outcome.
Okay, so we've got a fairly simplified picture here, but it's more or less accurate for what we want to present. But effectively, we've moved from the cell division, so more of a reproductive phase of the tree, into the growth phase of the crop. So what we're now doing is using nutrients, water, and sunlight and carbon dioxide effectively to produce the what we call as secondary metabolites. So the sugars, amino acids, proteins, fats, all those compounds that actually make up flesh and the seed inside the fruit. So when we talk about maximizing fruit size and maximizing the growth or the maximizing fruit size and maximizing fruit growth, effectively what we are really talking about is trying to leverage photosynthesis and maximize its production of all of these compounds which go into making up the fruit. So and also a side note, or quite an important note to realize, is that nutrients aren't energy to this cycle. So nitrogen, for example, is not energy to the plant. So when we're putting nutrients on, we are supplying the plant with the parts that it needs to complete this process in the most efficient manner possible. So ensuring that we provide the nutrients for photosynthesis rather than just trying to push the tree for maximum vigor and maximum growth, we can actually get large leaves and strong growth in the fruit without necessarily pushing shoot growth and uncontrolled top growth in the tree. So if we have a look at some of the key nutrients for photosynthesis, we have magnesium, nitrogen, and both of these are used in the actual makeup chlorophyll, in the makeup of chlorophyll. Iron is important for the production of chlorophyll. So we need to have iron there to be able to utilize these other nutrients. So this is where it becomes more and more a balancing act between the nutrients, as
I'll just do that anyway, actually.
Compared to just more of
I'll just put the
one
combos
particular
behind
nutrient
those.
or two particular nutrients is better. So we need to ensure we have the iron, and then manganese comes up again as being part of a process that we know as water hydrolysis, which is the very, very first step that happens in photosynthesis. So where the water is actually taken and split and utilized in the process. So if that manganese isn't there, that first stage of photosynthesis doesn't happen, and it doesn't matter really how much of other inputs you might put on. If the plant can't photosynthesize or can't utilize its water correctly, all you do is increase the stress on the tree and compromise the quality of the fruit effectively. The other one here is phosphorus. So it's important most of us have good levels of phosphorus in the soil, but it is just important to be aware of phosphorus level in the plant and whether it's adequate enough because it is critical in bringing some of those other nutrients into the tree and transferring energy around the tree due to its relationship or its integral part of ATP, which is the energy transporter and fuel of the plant effectively. So the main key is to have these in balance in the tree, and you can manipulate that these days, or actually test that these days with some of the newer sap testing methods that are coming out, which give us more detail as far as what the ratios of each nutrient are in the tree, and less susceptibility to being affected by residues of chemicals and other products that might not actually be designed to go into the tree. So then we have some other key minerals which build on top of photosynthesis which are required for cell expansion. So we've got the ones which are for photosynthesis, and then these are probably more utilized in the cell expansion phase or process in the tree, in the fruit, sorry. So we have potassium, which is, potassium is in quite a high level in the fruit. Actually, minerally, the fruit of a cherry contains about 1.2% potassium. So it's almost a 1-to-1 with nitrogen. So it's actually quite a high removed nutrient that we actually take off the farm each year. Obviously, we need to keep that up, keep that calcium and boron supply to the fruit, and then copper, again, just to ensure that we have that level of copper there, which is important for our skin elasticity to ensure that we can have the expansion and contraction of the fruit as it requires. If we move on to ripening, so as the fruit ripens, it's actually going through another whole, quite a complex process is actually happening in the fruit as it's ripening, and some interesting studies been done on what nutrients have, or what nutrients can affect what's happening at ripening. And it's two examples that are interesting to me. Is boron has actually been found to bring on the maturity of the fruit. So higher levels of boron at this stage will actually speed up maturity, and then that's to do with its relationship with ethylene production. And then cobalt is another nutrient that has been found to actually slow down or delay maturity in various fruits. So there's some interesting work that's been done in nutrient relationships with ripening. But probably one of the more important parts of during ripening and harvest is bud initiation is taking place. So this is where all of the buds for the next season are actually beginning to initiate in the wood, and it's probably quite a lot earlier than what a lot of us give it credit for, but it's is quite a critical stage to ensuring we can maintain that crop or have that crop load in the following year. And the main aim at this stage, really, or anything that we do during this ripening stage, very closely after harvest, should be based around minimizing the stress on the tree, because it's going through quite a complex process here where it's beginning that new bud development. And if we can minimize the stress on it, we'll have more bud formation for the following season, and which gives us more options when it comes to pruning and how we might want to set up the crop for the next year. So probably some of the key nutrients which are relevant at this stage, and also keep in mind that this is actually after harvest as well. So it just goes slightly after harvest. Probably the traditional delay that we have between harvest and post-harvest applications of nutrient is probably not ideal as far as the tree is concerned, because we want to try and ensure the tree has everything to do that, to complete that bud formation process. So we've got boron, cobalt, which I've just discussed there more. They're just based on their effects that they have on the production, on the ripening. We need to ensure we have magnesium, sulfur, and molybdenum, all key parts of the nitrogen cycle in the plant. So where we really want to see nitrate levels. So we test nitrogen in a couple of different forms. We actually look at nitrate and ammonium and total nitrogen, which includes your amino acids and proteins. And what we've found is we get the best results as far as fruit quality when we actually have the lowest number of nitrate, not lowest concentration of nitrate, and higher concentrations of total nitrogen. So these magnesium, sulfur, and molybdenum are very, very critical for the conversion process in the plant, which takes that nitrate that we might apply, or ammonium that we might apply, and converts it into those amino acids and proteins, and it ensures the plant is actually able to utilize it in a healthy way. We've got Andrew's just quickly going to, I think, summarize some of the things we've gone over, and then we'll just see if anyone has any questions, or we'll go from there.
Okay, thanks. All right, so when we need to look at managing effective nutrient applications, as Joe's mentioned, we need to maximise the fruit growth, which is an actual outcome of the photosynthesis cycle. So if you don't actually have that photosynthesis cycle functioning properly, you're not going to get your best outcome based around fruit growth or shoot extension. So, you know, that's really the key take home from today is photosynthesis is the first step, and the rest of it is the outcome of that. So, you know, the key things around that is the trace elements are really critical in that stage. And as Joe's mentioned, deficiencies in that particular stage can create a major reduction of our photosynthetic cycle or sugar production, protein production. So we really want to maintain that correct balance of nutrient applications to try and minimise the plant stress. So you probably, for those that have been around the industry a while, have sort of heard guys like Kyle Matheson say, you know, happy leaves, happy trees, good fruit sort of thing. So if we can see that good, really big, strong leaf, we know that we're going to maximise that sugar factory, which we've got producing in the background based around the photosynthesis cycle. So we move to just our last slide, which will sort of sum up what's going on through our various stages, which we can actually have an impact on. So we talk about the things we can control as growers, but we know we can't control the weather, but some of this staging of nutrients, which as we go through these sort of stages, we can certainly have an impact of what we can do. So you need to know what's going on in your plant, and that's when your guys that are helping you on the ground, your agros or whoever you're working with, can hopefully direct you in where you need to go. But during that cell division stage, we're in that strong reproductive state, so we've got to make sure that calcium's building that structure and integrity of the cell wall, which is pivotal for the cell division stage. Calcium requirements also happen much earlier in the crop than we previously thought. So, you know, through that bloom cell division stage, you know, early to 100% bloom, as we've spoken about earlier, we've got that 50% of our cells already formed, so let's make sure that calcium's available. Manganese, once again, is regulating that potassium movement, so we're trying to keep that calcium environment happy through that cell division phase with manganese. And then once we get to the cell expansion, we move into that stronger growth state. So once again, we're working with our photosynthesis because it's the key driver for the energy in the tree. So it's ensuring that correct combination and balance of nutrients maximise that efficiency. An excess of nutrient will impact the stress, which is a negative outcome for our fruit quality. And then once again, once we get into that ripening, the nutrient applications need to be, once again, minimising that stress, particularly because we've got that bud initiation process which is going on. And you hear how people have that roller coaster in sometimes biennial cropping. It's because we're putting the tree under stress when we're actually just at that critical harvest stage. So, you know, nutrient management really should be targeted all the way through that particular crop cycle. And we as growers really can influence that if we can just make sure that we're maximising that photosynthesis cycle, which will give us the best outcome for our fruit, particularly along that journey and at harvest, because once we get the fruit in the box, that's where we want it to be. Have that good fruit integrity and quality that we can, put in a box and know that our customers can enjoy, because that's what it's all about. So that's pretty much it from us. So probably hand back to you, Tom, and if there's any questions that anyone would like to for us to answer, or if we've answered all the questions as we're going along, all well and good. But apart from that, I'd like to thank Cherry Growers Australia for putting this on and being a part of it. And if you need to know any more about this particular information, just contact us either through Cherry Growers or ourselves. So yeah, more than happy to help out. Thank you, Tom.
Tom's had to leave, so thanks Andrew and Joe. That was a fabulous presentation. Took me back to Biology 101. Can't see any questions in the chat at the moment. Does anyone have any questions?
Yeah, I have a question, just probably for the hybrid fellas. How you going? Just in your slides, there was lots of key elements at key times. I wasn't clear on nitrogen or nitrate, or whichever form you want to talk about, and what the key times on the different nitrogens at specific times.
Okay, so nitrogen obviously is a very critical element, but what we're trying to get is, obviously most of the nitrogen that we apply becomes in either a nitrate, so calcium nitrate, potassium nitrate, or ammonium sulfate, for example, just some common applications of nitrogen. And the key the whole way through the crop is actually to get that converted into usable nitrogen, which is usable and functional to create proteins and amino acids. So to do that, we need to ensure that we have any nitrogen application is going with those other nutrients that are mentioned there, magnesium, molybdenum, and sulfur, for example, as some of the more key ones. And then basically the other nutrients are just parts of that photosynthesis cycle. Is that what you're looking for, or is there some further?
Probably just to add to that is, I think based around timing is you've got to make sure that you pass that cell division phase, because we're trying to push the tree into a hormonal shift from cytokinin dominance to more auxin, and typically nitrogen, depending on what form you bring it in, will be a growth element. So once you've got into that phase, that's when you can apply your nitrogens. But I think the key thing that we're trying to really ram home here is that if you can convert your nitrogen or nitrates into your proteins or sugars, you'll have a far better piece of fruit than having a piece of fruit that's loaded up with nitrate as such, which is not converted, because as we know, that will impact fruit firmness, and it'll also give us a headache once we get to a thunderstorm or a rain event, because we'll already have a tree which is loaded up in nitrate as such. So we've got to be able to maximise the efficiency of the conversion while making sure those trace elements are available to help convert the nitrate to our proteins.
Yep, so we have got another question, Andrew and Joe. Is it better to apply foliar nutrients or fertigate?
Probably both. So depends on what you're applying. So basically, the cheapest form of nutrients generally is actually dry forms, so granule spreads and that kind of thing. Then we generally will use a combination of fertigation and foliar, depending on what we're trying to achieve with. So if we're using trying to get calcium into the plant, for example, during that cell division window, we'll actually be fertigating quite a lot of calcium. And then if we're later in the season, if we're trying to get calcium in, we'll be foliar spraying quite a lot of calcium. But probably the key with foliar nutrition is to ensure that it's in a chelated form. We typically find very minimal results from using straight sulfates or oxides forms of trace elements particularly. So trace elements we typically will foliar spray because we need them in much smaller amounts, and you can get very, very low rates of chelated trace elements that are very effective. Probably some more of the majors: nitrogen, potassium, phosphorus, calcium, depending on what stage of the crop we're at, will be probably more fertigating it because it's more, and also that's based around the cost effectiveness of it too.